quickly, I'm going to recap enzymes for you. This is probably not one of the areas you're concerned very much about, but if you're watching all the videos, I appreciate you. There's only 7 slides I gave on enzymes to begin with. Biggest thing about it, enzymes are almost always proteins. I'm not going to say always because that's not true, but for our purposes, they're proteins. You need to know that. You need to know why that's important as well. In general, enzymes promote chemical reactions within cells. It does not mean, and I'm going to put blinking lights on this as I say it, chemical reactions can take place without enzymes. They can 100% take place without the presence of an enzyme. An enzyme is there to catalyze a reaction. Catalysis is speeding up the reaction rate of a chemical reaction. It does not mean it's causing it to happen. It's speeding up the rate at which it does happen. Oh, I hiccuped. All right. Now, how it does this is when you're catalyzing a reaction with an enzyme, you are lowering the activation energy required for the chemical reaction to take place. So going back to like middle school science and biology, there's a certain amount of energy required. I talk about it as rolling a rock up a hill. To go have the rock roll down the other side, you have to get to the top of the hill first. If the hill's really high, it'll take longer. If it's a shorter hill, it'll go faster. That's the idea with an enzyme. Enzymes create the shorter hill. So it lowers your activation rate so that you can increase the rate of the actual reaction. Now, how it works is that an enzyme has its one perfect match. It has its substrate. Now, substrates can be, they're usually smaller than the enzyme. Remember, the enzyme is a protein, so it's a little bit more complex. Substrates typically are like sugars and fats and things like that. Not always, but in general, smaller substances. Now, it doesn't have to be one substance that breaks apart. It can either break things apart or put them back together. And what I mean by that is like with lactose, it gets broken down by lactase. Its enzyme is lactase. Lactose starts as a disaccharide from our macromolecules group. Now we know when lactose goes into our body, it's supposed to break down into its 2 pieces of glucose. Okay? Now, when it breaks down, you now have glucose, which is the usable form in your body. Think about it. What's the very first thing that goes into cellular respiration? Glucose. And so for your body to be able to use it well, you can't process this big sugar as it is. It needs to break down first. And what lactase does is break that apart to speed up the chemical reaction. And we talked about being lactose intolerant. It doesn't mean that you can't break it down. It's not that you're allergic to it. It just means it's really difficult for your body to break down big substances. So enzymes are there to be a helping hand. That's the idea behind it, to kind of draw the different pieces that we've been using in together. Now, the substrate part of it, remember the enzyme seeks that out. There's a perfect match, and what we use to describe this is a lock and key or a Cinderella slipper or a puzzle piece or however we want to describe it. Basically, there's one perfect match. Now, what that means is that every type of enzyme has one type of substrate. It doesn't mean that an enzyme can only be used once and then never again. It means lactase, so with ASE being our enzyme, only works for lactose. Okay, so those are the only 2 to go together. Now, once it breaks down that substrate and releases it, it can be reused for our enzyme. It doesn't mean one as in one substrate ever happening. It means one type of substrate, which I feel like I need to explain a little bit further for you guys than I did in class. Now, that also means that for the hundreds of chemicals we've got in our body, we also have specific enzymes that match those. So each one is a little bit different. Now, for our substrates, the enzyme seeks out the substrate. Once it finds that it has its perfect match, I told you, even if one of the other teachers put their key in my door, technically, mine should be the only one that turns it. Now, it might fit in the door, but it shouldn't turn the lock. My key should be the only one that actually turns the lock. It doesn't matter how many times I put my key in there, it will still turn the lock, and that's kind of the point I was getting at. That as long as it's my 100% perfect match key, it'll always turn the lock. If it's just kind of close, it won't turn the lock, and that's where that lock and key mechanism comes from. Now, when your enzyme and your substrate meet up, they create an enzyme substrate complex. Now, the actual shape where it binds and creates that temporary bind. It's called the active site on your enzyme. Now, one of the parts of the video went over that I am not going to include in your test, but just so it doesn't confuse you, I'm not forgetting about it, is induced fit. Basically, the enzyme is going to try to hug the substrate and pull it in as tightly as possible to carry out the reaction and cause whatever it needs to happen. So again, I'm going to go back to the lactase thing. Lactase will bind with lactose. It'll give it a big hug. It'll break the pieces in half and then move on. Okay, so that's its purpose. It's trying to carry out that reaction and speed that up. Once it does that, the enzyme is unharmed. So remember, for some of my classes, I gave you the weird example of the purple bear. It gives a big hug, and the bear is big and strong, so when it hugs the substrate, it comes unscathed out of it. It's totally fine. Nothing's wrong with it. Now, the substrate has either been morphed into something else or broken apart, or it has some sort of change in the shape, the substrate. The enzyme keeps its shape. Now, why that's so important for us is because it's a protein. We know the shape of a protein is referred to as its conformation. We know when it experiences a change in its shape, that's called denaturing, and we know that damages our protein. Now, think about why that's important. With an enzyme, we know it has one perfect match, and that's based on the shape that it is. Now, if it loses its shape, it loses its functionality. It no longer has a perfect match. Now it's just kind of a big pile of goo that we can't really use. So when an enzyme denatures, that's not because of the actual bind between substrate and enzyme. Remember, when that happens, it hugs it, does whatever it needs to the substrate, the enzyme is still usable. Enzymes denature, their proteins denature, because of outside sources, so something happening to it. Now, examples I've given in the past are high temperatures, pH. I told you on your PowerPoint that if temperatures are low, so like it usually functions between 45 and 55 in the body. If it's below 45 degrees Celsius, it'll go into hibernation. Now, if it hits extreme lows, it'll denature. It will also denature if the temperature is too high. Okay, it doesn't go into hibernation. It just, think about it as boiling. Think about when your body gets a fever. I've said this a bunch of times. If you have a fever, it's okay for a certain point, and then when it gets too high, you go to the hospital. It's because your body is literally going to start shutting down because it's not made to function at that temperature. Our bodies like to stay within a pretty narrow range of body temperatures. We know when our body temperature is too high, something's wrong. We know if it gets too high, something's really wrong. We have to fix it, or it's lethal, to be honest with you. It doesn't happen often because our bodies like to stay in that range. So it does cause denaturing. Other things that cause denaturing, radiation. So thinking about like with our cancer topic, cancers we treat with radiation. Some of them, not all of them. We talked about all the differences and things that go on with it. But we treat with radiation because the idea is that if we can treat with radiation, potentially we can break down proteins of targeted cells that are mutated cells and hopefully keep them from growing by denaturing them and damaging their ability to grow. So all of this should start to kind of piece together, and all of it should have overlap. I promise I don't just pick out these random topics to confuse you. They do go together. They do have different interconnecting pieces, and as we go through the year, it'll make more and more sense. Last thing I want to tell you about is that enzymes can be inactive, not because they're denatured or deformed or anything like that, but just because, I don't know, sometimes they're difficult. I gave the example of, think about a CEO that runs a company. That's all great and good, except they probably have a secretary, because as much as you could be an amazing human that does all this stuff, and you could be the smartest person on earth, there's only so much you can actually do for yourself, though. So a lot of very, very high-ranking and important or smart people have secretaries because they can't be there to answer their phone constantly, schedule calls, check their calendar. They ask someone to help them out with that. Not because they're not capable of doing it, because sometimes you need a helping hand, or maybe you're lazy. I don't know. But when we're talking about enzymes, those helping hands, those aids, are non-protein components called cofactors and coenzymes. Now, the only difference I need you to know is a cofactor is a lot smaller. It's a molecular level, whereas a coenzyme is an organic molecule that's a little bit bigger. So going from ions for your cofactor, which is small, and then putting a bunch of that together to get a coenzyme. And then again, drawing more connections between all our different lessons. we talked about cellular respiration, and acetyl-CoA is acetyl coenzyme A. So just keep that in mind, that there are a lot of different pieces you're going to start to see, like, oh, I remember learning about that. Now I get why we're doing it. If you have questions about this, please feel free to email me. I will be on my computer pretty much constantly over the next few days, and definitely over the weekend for any of my students taking it on Mondays. So I'm hoping these are helping so far, and we've got one more to go.